Hi guys, welcome back. Video number 2. In case you're trying to catch up with biology, trying to cover a lot of stuff in a short amount of time, this is what we're going to try and do today. So, the topic of today is light microscopy. Now, what are the 3 essential questions? I'm hoping there's always going to be 3, but I don't want to force 3 out of it every single time. But what are the 3 questions? Number 1, how are samples prepared for light microscopy? Okay. 2, how do we calculate magnification? Always everyone's favorite. Okay, how do we calculate magnification? How are eyepiece graticules used? Number 1, how are samples prepped for LM? How are samples prepared for light microscopy? And in this case, what we're really thinking of, so the first concept, right, and it's always like central concept, and then what does that lead to? Central concept is that because we're using visible light, and this is transmitted, visible light is transmitted. So in order for us to see anything in a light microscope, it really needs to go through the sample. So because we're using transmitted visible light, it needs to go through the sample. And for it to have any chance of being able to go through the sample, it needs to be thin. Okay? But thin on its own would not be great, because if light just goes through, we won't see anything. So we do need contrast as well. So we need thin, and we need contrast to be able to see the different parts of the cell, the different structures, be able to differentiate structures, identify structures, and so on. So thin and contrast is needed. And now let's look at the different types of sample preparation, which should incorporate these things. Okay, how are, so first one, we have the dry mount. Okay, so your sample, thinly sliced with a scalpel, thin slice of it on the slide, place the cover slip on top, and off you go. Dry mount. Okay, and you'd usually use the dry mount for things that are not living. Okay, the other one is you've got a wet mount. So you might have you might have a sample, maybe a bit of a plant. It's got living cells, and for them to kind of maintain their structure, their functions while you're viewing them, you might need to put the water on the sample, around the cells, around the tissue, for it to maintain its structure and so on. Okay, so you've got a thin sample. You might have even aquatic organisms. So the wet mount, you might have living organisms, aquatic orgs, right? You might have living tissue, such as plant. Now, in that case, you want to have that sample, put it on the slide, put a drop of water on it, and then place the cover slip on top, and then you view your sample. Okay, and in that case, you see the real colors because you haven't added any stains. All right, and it's thin. Either the sample is very small, so it's thin, or you've cut it very thin with a really good scalpel blade carefully, and so on. Right, and then we have got squash slide, right? So in a squash slide, you get a sample, but then and if that sample is bulky, thick, in that case, what you want to do is you want to separate the cells from each other. So when we use this is like a root tip squash, for example. So when we want to see mitosis or observe mitosis, we've done a root tip squash, and what we take is we take a root, but the root is, it's three-dimensional. You cut that section off, it's three-dimensional, and that's not going to be thin enough. So what we do is we add acid, we add hydrochloric acid. It kind of dissolves the kind of glue that's between the cells, and then when we place the cover slip on top, it flattens all the cells out because they're no longer stuck to each other. It makes a single layer of cells that we can more easily observe. Okay, so we've got a squash slide, and there's a fourth one which I can't remember. Dry mount, wet mount, squash slide, and we've got a blood smear. So smear slide. So you take, usually this is for blood smears, right? So blood drop, and you use the cover slip to kind of spread that drop out in a very thin layer, and gently press the cover slip on so that it spreads all those cells out. Remember, those cells are not stuck to each other because they're in the blood, they're in a liquid, and it spreads them out, so it makes a thin layer that you can easily view. So those are the different ways that you can, or the different slide preparation methods. We need to also include in this differential stains. Now these are pigments, these are chemicals that have certain properties, and because of their chemical properties, well, one thing, they are always colored. Okay, so they are pigments. They might be red, they might be blue, they might be purple, whatever it is, but they have a color. But because of their unique chemical properties, they also tend to bind to certain things in the cell. Okay, so if it's positively charged differential stain, it's going to bind to DNA, which is negatively charged, and so then we can use that to highlight the nucleus or the DNA in the cell. And there are other things which might have other properties and therefore are binding to other components in the cell. But what they do, because we know what they tend to bind to, we can stain the cell, we can add the differential stain to the cell. The differential stain goes into and onto the cell. It binds to the specific cellular component and then allows us to see that structure by increasing the contrast. Okay, so it is false color in a way. It is false color because it's not showing the real color of the thing, but it does increase contrast. Okay, it does increase contrast. It allows us to see specific structures or parts of the cell. That allows us therefore to identify cells, identify cellular components, and so on. Okay, so that is all about slide preparation. We move on to our second question. Magnification, right? How is magnification calculated? Now, look, if I'm honest, this question itself can be like a full video. It can be a full lesson, in fact, because it's mainly you have to practice doing it in order to get good at it, right? However, when you're revising, it can still serve a purpose to go over this stuff. All right, so how is magnification calculated? What are the bare essentials of what you need to know? Okay, it's amazing how many magnification questions come back to the formula triangle. Okay, so remember that the image is produced by the detection of the emissions which have been magnified. But the emissions came from the actual object or sample itself. So the actual sample was excited, right? The emissions came off. We refracted or diffracted those emissions, which resulted in the magnification of the actual size of this thing, resulting in an image which was much bigger than the actual. Okay, so we have these things. We have the image, we have the magnification, and we have the actual. Okay, now this seems straightforward, and I'm sure you're nodding along with this, or maybe you hit the skip button already. Okay, but the thing is that this part is easy, but when you get an exam question, it's not always obvious which one is which, and I think that's when the problems occur. So I might not do anything more than to just give you a few clues as to how you identify these, and I would incorporate this into your revision so that it just reduces the load on your brain to have to figure these things out in an exam question. You can then rely on your routines and methods. Okay, and you can practice that along the way to see if it works for you. Okay, but I think too many students kind of, they do get this and then they're just relying on this, like, I can deal with it when I get the question. Rather, you need to kind of make routines to make this knowledge more effective for you. Okay, so magnification should be a large number. How the actual, right? The actual is something that you know is too small to be seen. Okay? I'm just going to leave you with that to think about. Your image is something that you can measure with a ruler. So if you're putting your ruler down and measuring something, automatically that technically should be your image value. Okay, there's one other ingredient to this process which we need to remember. Units. Units, okay? Now, your go-to unit in biology should be the micrometer, okay? You're usually measuring in millimeters, okay? And yeah, so you're usually measuring in millimeters, and your go-to unit is the micrometer, because things in biology, like substructures in the cell, are usually on that scale, okay? So just remember that the conversion between millimeters and micrometers is 1,000. Okay? And that there are 1,000 of these in one of those. So to convert micrometers to millimeters, you need to divide. So if I'm going that direction, I need to divide. But if I'm going in that direction, I need to multiply. So if I want to see how many of these are in those, there's obviously going to be a lot more. I'm going to multiply that by 1,000. Okay, and I think for most questions that would cover it. Okay, now let's move on to our third question. How is the calibration involved in all of this? Used. How are eyepiece graticules used? We're going to do the quick version of this, remember, because this should be technically revision. If you want to get into the nitty-gritty of this, I suggest you pause here and go do that research if you need to, and then come back later. Okay? But the quick version of it is like so. The eyepiece graticule, its purpose is to be able to estimate the actual size of samples being viewed. All right, so if you're looking under the light microscope and you're looking at a xylem vessel, the presence of the eyepiece graticule should allow you to see, to allow you to estimate how wide that xylem vessel is. Okay, so let's say I am looking down the sample and I'm looking at my cells. Okay, right, of course, some cells. It's a terrible biological drawing. Okay. All right. Now, in the eyepiece, right? In the eyepiece, you will have your eyepiece graticule. So this contains the eyepiece graticule. It's right here. It's printed on the eyepiece lens. Okay, or it could be a sticker that's stuck onto it. Now, because of that, you'll get a grid or a little thing, little scale that is in front of everything that you look at. But because the magnification is happening due to the objective lenses here, right? This scale has no units. So we don't know, even though I'm looking at this, I might say, oh yeah, that's 1, 2, 3, 4, 5, 6, 7, 8, 9. That's 9 graticule units. That xylem vessel is 9 graticule units wide. But I don't know what each graticule unit, what is that in actual distance. I put the stage micrometer there, and the stage micrometer might look like that. The stage micrometer is always 1 millimeter, and there's always 100 divisions, right? So there's 100 divisions here on the stage micrometer, and they represent 1 millimeter in total, so from end to end. So what I can say is, right, there's 100 divisions here, and there's, each of these is 0.01 millimeter. So if each of these is 0.01 millimeter, if this lines up, then one division is equal to 0.01 millimeters. So when I then look at my xylem vessel, I can then estimate how big that xylem vessel is, because I know now that one division on the eyepiece graticule is equivalent to 0.01 millimeters. Okay, so that's what calibration is, right? Calibration allows us to estimate the actual size of a sample being viewed, okay, by allowing us to calibrate, right, the length of eyepiece, graticule, divisions. So once we know how big eyepiece graticule divisions are, that's what the stage micrometer is for. So once that gives us that conversion factor, then we can use that conversion factor to estimate our samples. Okay, so let's recap our essential questions. This is, at this point, the easy stuff is done and the hard work now begins. You need to be able to answer these essential questions. Okay, so essential question number 1, how are samples prepped for light microscopy? Okay, question 2, how is magnification calculated? Okay, yes, most of your work here is going to be practice. It doesn't hurt to just recap that theory and just know why the formula triangle is the formula triangle and so on. Remind yourself of the units and how to interconvert between the units. That's revision. Sorry, that's retrieval. Practice that retrieval. Okay, it's like working a muscle, right? If you don't work it, it doesn't get stronger. 3. What was 3? How are eyepiece graticules used? Answer these, okay? And farewell. Right, guys? So get practicing. These are your essential questions for this focus on light microscopy. There's a lot of practical work and a lot of maths in this section, but we can still revise it, we can still retrieve it, we can still practice it, because then it strengthens those connections to our practical knowledge and our mathematical confidence. All right, guys? Good luck.